Hi everyone, thank you for coming to our second Everhart Lecture Series Talk of the Year. My name is Rachel Tam. I'm the Academics Chair of the Graduate Student Council and also part of the Everhart Lecture Series Committee, along with Pan and Surya. And so the Caltech Everhart Lecture Series is sponsored by the Graduate Studies Office and organized with the Graduate Student Council. The Everhart Lecture Series is a forum for outstanding Caltech graduate students to present their path-breaking, interdisciplinary research to a broad scientific audience. We're excited to continue the lecture series after about 5 years, and so I will now like to ask Professor Gutman to introduce today's speaker, Dr. Prashant Bhat.
Okay, well, thank you all for coming out. It's a great pleasure and honor for me to get to introduce Prashant to all of you, who many of you already know, our guest speaker who will deliver this Everhart Distinguished Graduate Student lecture. Prashant is currently an MD PhD student in our joint UCLA-Caltech Medical Scientist Training Program, which means he is going to be both a PhD and an MD when he finally finishes his training in the next few years. When he started his career, he started at Berkeley as an undergraduate, received a BA in molecular and cell biology, where he worked with Jennifer Doudna, who was a pioneer in the development of CRISPR. In fact, won the Nobel Prize recently for her pioneering work in this. And even before CRISPR was a household name, Prashant was working with her on some of the very early characterizations of many of these different CRISPR enzymes in bacteria. After finishing his undergraduate work, he came to Caltech, to UCLA Caltech, did some medical school work, but we'll skip over that for a moment. But as a PhD student here at Caltech, he's been in our biology program, and I've had the privilege of working with him for the last 6 or so years, which has been really, not to make this about me, but really wonderful for me, and been really wonderful opportunity. But what he's done is really incredibly remarkable. His work has really uncovered some fundamental new mechanisms of how cells work and how three-dimensional organization of molecules in the cell can control fundamental information flow in the process of mRNA splicing. I'm not going to tell you about this because you'll learn all about this in today's lecture. But what I want to highlight is sort of this new and exciting this exciting new paradigm in cell biology that Prashant has uncovered, which really required, as was alluded to about this lecture series, required real interdisciplinary work in developing new cutting-edge experimental approaches, analytical methods, and working with many different colleagues across many different divisions here at Caltech. In recognition of his many important contributions, Prashant has been the recipient of several prestigious awards, including the De Carmen National Fellowship, the Ruth Kirschenstein National Research Fellowship from NIH, and not least this Everhart selection for this lectureship, and amongst many others, and certainly many, many, many more honors to follow. Prashant has also demonstrated an important commitment to diversity and inclusion. He has played an important leadership role in the Caltech PRISM community and is a recipient of the Caltech Diversity and Inclusion Award. So please join me in welcoming Prashant, and we're delighted to hear about all your amazing work.
Okay, can we hear me in the back? Okay, well, thank you so much, Mitch, for that introduction, and thank you to the Everhart Committee and the Graduate Studies Office for giving me the opportunity to present my work today. So I have 2 parts of my talk. The first part of my talk, I'm going to be talking to you about how we developed a new method from biology to look at the highest resolution picture of how three-dimensional molecules organize in space, and we did this without ever looking at a microscope. And the second part of my talk, I'm going to then take these maps, and we're going to ask the question of why molecules are organized in this way. What makes it that these molecules are arranged in three-dimensional space? What does that mean? And so the structure of the talk is pretty simple. So the first part, I'm going to be talking about structure. So we're going to be making molecular maps together. And then finally, and then we're going to go into the functional implications of this. So like, what do these maps even mean? And then finally, the applications of these findings to clinical medicine. And to get everyone oriented on the same page, I want to introduce you to the biology that I think of and how we think. And so this is the fundamental principle in biological systems, and that is the concept of structure and function. So this is not only biology, but in all aspects, like when you're at a symphony, there's structure-function relationship. How? Well, when you're listening to like the wind, the percussion, the string instruments, each and every single instrument that you hear has a different shape, and because of that, it makes a unique sound or its particular function. So in a similar way, biological systems are no different. So here we have different organs, like there's tiny little alveolar air sacs in our lungs that help us breathe. There's also in our colon little appendages that help us reabsorb water, and then our stomach, and its structure is very important for the digestion of food. So in all of these cases, structure and function are very important. So what happens if we go many layers deeper? Well, humans and all biological organisms are made of cells. Humans are made of trillions of cells, so your lungs, your colon, your stomach, and everything in between made of cells. So if you took a microscope and you peered down under it and you took a cell and you looked at it, it would look something like this. This is a schematic of a cell, and what I'm showing you here is that here in the cell there's a membrane. The membrane is like the skin for the cell. It keeps the outside out and the inside in. Now what happens if we go inside, past the membrane, into the bellows of the cell, we're now entering the cytoplasm. So cyto means cell, plasm is gel. So it's like a jelly-like substance that keeps a bunch of structures together. So what are these structures? Well, these are called membrane-bound organelles. So these go by many names, and I'll just highlight a couple examples. One of them, like the mitochondria, and this is where the energy is produced in the form of ATP. So every muscle you contract or every blink you take, you expend energy in the form of ATP, and it's made in the mitochondria. Additionally, we have membrane-bound organelles like the nucleus, and this is where our DNA is housed. So you get one copy of chromosomes from your biological mom, one from biological dad, and it gets stuffed in this organelle called the nucleus, and it encodes a genetic blueprint in order for you to look how you look, feel how you feel, and think how you think. Now, the title of my talk was about membraneless organelles, and so what do I mean by that? And in order to explain that, let me tell you what a membrane-bound organelle is. So a membrane-bound organelle, so let's take the mitochondria for example. Just like the cell, which had a skin or a membrane over its entire surface, mitochondria and other membrane-bound organelles do too. So what this would look like, and the names are not important, but this is what the membrane looks like. It's a phospholipid bilayer. And all that you have to realize is that it keeps the outside, the cytoplasm out, and the inside of the mitochondria in. And in this particular case, it's important that these molecules are not moving out, and it's known as restricted molecular exchange. So molecules are not moving out of the mitochondria. So what about the cell nucleus? Well, the nucleus itself is a membrane-bound organelle, but for the past century, there's actually a different type of organelle that has been really elusive to molecular biologists. And that is, there are these membraneless organelles that decorate the entire nucleus of the cell. So these are membraneless organelles in that they don't have any enclosing membranes, but they still have an inside and an outside. But in fact, when you look, there's like this invisible boundary, and it's a question of like, what are keeping these molecules together? So these represent distinct entities with an inside and an outside, but in fact, there's free molecular exchange into and out of the cell. And there are many such examples, so let's make this a little bit concrete and provide some examples. So if you're a biologist or took a biology class in high school, or I'll explain it to you right now, there are many different membraneless organelles, and the most famous example of which is the nucleolus, plural nucleoli. And there are many others, like speckles, which I'll be telling you about later in my second half of my talk, Cajal bodies, histone locus bodies, there's so many of them. And the basic structure of these is that they're organizing molecules around shared molecular function. But even though that we know that they have shared molecular function, we actually still don't know why they exist, and that we don't know what the functions of these nuclear structures are. So that's a little confusing, so let me explain that once more. And that is, if I were to take the membrane, so basically, well, I told you about these structure-function relationships, and so now I'm saying that, well, maybe we don't understand the functions of these things. And so you might remember from high school biology that the nucleolus is the site of ribosome synthesis. So the ribosome is the machine that actually makes the proteins like collagen and keratin and insulin and things like this. It's made by the ribosome. But the ribosome has to be made somewhere, and it's made inside of the nucleolus. But here's the catch. If I were to take a mitochondria or a membrane-bound organelle and rip off its membrane and put its contents into a test tube, it would not work. So mitochondria would not be able to make energy. It would not make ATP. Now, if I were to do the same thing with the nucleolus, where I took the contents of a nucleolus, I put it in a test tube, I shook it up, I would still get a ribosome. So that's a little confusing. So now Prashant's telling you, does structure not equal function? So this is the conundrum that we're in, but of course, all of these molecules are organized within these very beautiful structures, and we really want to understand why. And today I'm going to tell you why. But before I tell you why, I want to walk you through how we even got to the point of addressing this question. This was a question that has been throughout the past several decades of like, why are molecules organized in this way? And people have had hypotheses, but experimentally proving it has been a little difficult. And for that, I want to take you a little bit back into the 60s and 70s when people were actually looking at where genes are being mapped. And I have to talk about this because modern technology has enabled us to make these technologies that enable us to understand why are membraneless organelles doing what they do. So again, let's make this concrete. So we get one copy of chromosomes from our mom, one from our dad. And so we have 22 sets of chromosomes, which is what you see here, an X and a Y, Y in males, and two X's if you're a female. And so it was painstaking to map where genes were at the time. And that is, if you were interested in a gene like hemoglobin, it carries oxygen in your blood. So that seemed like a very important gene to know where it is. And so it was found, okay, it's on chromosome 11. And so, people brute force went through, and then it was keratin. Keratin, there's a bit on chromosome 12, a bit on chromosome 17. But of course, we have thousands of genes that make up our human genome. And so this is naturally a very difficult thing of mapping every single protein inside of the genome. So how do we map all the genes on every single chromosome? Well, this is where the Human Genome Project came into being. And so this was started in the Clinton administration and completed in 2000, in which we sequenced the entire 3 billion nucleotides of the human genome. So scientists took the nuclei of cells from humans, they took out the DNA, it's 6 feet long, it gets packaged into this tiny little space of the nucleus, and it's made up of these letters A, T, G, and C. And so from that, we got every single chromosome, every single letter, we can read it from end to end. And I'm going to just highlight a few little nuggets of information that was learned from the Human Genome Project. And that is, one of the surprising things was that only 1% of the entire genome actually codes for protein. And a very large proportion, 70 to 90% of the genome, the DNA is copied into its sister molecule, or RNA. And we're now just starting to understand what these RNA molecules do. Another surprising finding was that we share 99.9% of DNA. So me and you share 99.9% of DNA. You and Albert Einstein share 99.9% of DNA, and everyone. It's quite remarkable, and that the variation that we have is just the 0.1% that we see in DNA. And then another reason why people were really interested in the Human Genome Project is if we understood the book of life, the genetic code, then when typos arise or mutations in our DNA, we might be able to understand how to fix it. And so it was found that there's mutations that really occur in both coding regions that code for protein, as well as non-coding regions that do not code for protein. So this is all fine and dandy. And so in the past 10 years, people started taking a different approach. And this is around the same time that I started my PhD. Fun fact, this was when I was in like the 4th grade. And so we've come a long way since then. And so now, in the past 10 years, around when I started, when I moved to LA, there was a consortium that was started called the 4D Nucleome Project, 4-dimensional nucleome. And so it was trying to take the genome in 3 dimensions. So we had the string of letters, But now, how do we look at these string of letters in 3 dimensions? And so this is what they did. And we only knew a little bit about how DNA organizes in 3-dimensional space. So for example, there are some structures in DNA. So like this is a representation of DNA in normal cells. And it creates this loop structure where you have these 2 different DNA sites. They find each other. There's a protein that can bind to these DNA sites and stabilize this loop. So it kind of looks like a necklace. And if you think about this necklace, here is a pendant, and this pendant is a gene. And it's an oncogene. So we want this to be turned off. And in fact, in normal cells, right now, as we're sitting and breathing, there are these oncogenes that are just sitting there, but they're insulated. They're not being turned on. But sometimes you do get a mutation. And in cases like glioblastoma, brain cancer, where you get a mutation, this leads to disruption of the structure and loss of insulation, and therefore this gene turns on and you get cancer production. Not good. So obviously we have to understand how exactly these structures form and actually what these structures even are. Because I can actually count on 2 hands the number of examples like this that we know of. Many more are still to be discovered, and in fact, we developed a way that is going to be able to discover these structures. So how do we map all the molecular interactions simultaneously? I'm going to walk you through the solution that we developed, but before that, I'm going to do a little bit of chalkboarding of how, or whiteboarding, whatever you want to call it, how we actually determined what strategy to go down. So let's say I want to take a picture of this cell nucleus, and I want to look at region A, B, C, and D. So to give you examples, so region A has these hemoglobin genes, B has these genes that are encoding for keratin, C is the other chromosome that encodes for keratin, and D is something else. And so I want to take a picture of all of these things. And so what I could do is I could use a microscope, and I could add a fluorescent molecule on these different segments using technology I'm not going to really get into. But basically, you can label specific segments of DNA and say, OK, well, these 2 regions are in the same 3-dimensional space. Well, now what if I want to know where the keratin genes are? I want to know that they're far apart from each other and they're far apart from the hemoglobin genes. Well, we can add a couple more colors. But now it gets a little tricky because the disadvantages are, so at the time of when I started this project in 2016, we had a finite number of colors. You kind of only have the colors of the rainbow, which are 6 or 7 that you can choose from. So you can really only image 6 or 7 molecules at a time. There's another limitation in that the resolution is limited to 0.2 microns. And so to give you a sense, that is quite significant in the context of a cell, because if you have region A and region B and they're 0.2 microns or shorter distance near each other, in reality, they represent distinct areas of the cell. But when you look under the microscope and get your data, you're going to think that they were at the same place. So also not great. So what else can we do? Well, we can use this idea from sequencing. And because I know the actual sequence of these DNA molecules from the Human Genome Project, what I can do is I can isolate this complex. I can put it in a single little tube. I can sequence the DNA and get the exact string of letters and say, OK, well, all of these molecules were interacting in the same space. And I can do the same for B, C, A. And I can do this for thousands and thousands of different complexes. But there's a problem. I can't do 1,000 experiments because that would take me many, many years, and I only have so much time here. So we had to get creative, and this is the solution we developed. And this is a method called SPRITE, and it stands for Split Pool Recognition of Interactions by Tag Extension. Doesn't matter what it stands for, but I will walk you through how we do it. And so here, we have the same nucleus with complexes A, B, C, and D, and we want to know that they are all interacting in the same space at the same time. So the way that we do it is we freeze the cells with a cross-linker called formaldehyde. We then break up the complexes into smaller and smaller chunks. Then we pool everything into a single tube, and then we split it 96 ways into individual wells. And so the chances that a molecule lands in any one of these wells is 1/96. So we have, right now, we have complex A and B that ended up in the same well, complex C and D that ended up in the same well, and therefore they share the same tag. Well, this is good in that we can separate these complexes from these ones, but right now we still think that A and B are together. So that's not good enough. So let's do it again. So we can pool all the molecules in a single tube, and we can split again. And once again, there's a 1/96 possibility that the molecules go into any of these wells, but the probability that any two complexes end up in the exact same well is now 1/96 squared. So now A and B are in two separate wells, C and D are in two separate wells, all fine and dandy, and now we've been able to separate A and B. But of course, there's millions of molecules within the context of the nucleus, so we're going to do this procedure not 3 times, not 4 times, but 5. And so after 5 rounds of barcoding, we achieve 1/96 to the 5th power. That's 1 in 8 billion chances of two complexes being in the same exact well with the exact same barcode. So it's very, very low. And this is great because now we can use genome sequencing and we can identify these clusters. So now we have the known barcode string, and then we have the known genes that once we sequence, we can align it to a reference genome and say, oh, these are hemoglobin genes and they were together. And these were keratin genes and they were together. And they have a unique and distinct barcode from the ones that were in cluster A, and so on for C and D. So this is all hypothetical, but what did we find? What's the data set that we uncovered? Well, as you can tell, if you didn't completely understand it, that's fine. You can ask me later. It's obviously a very elegant, sophisticated way, but what the data we got was very elegantly simplistic in that the genome actually arranges in 2 distinct hubs. And so here is the blue hub, which is, so this is a spider plot showing different chromosomes, so chromosome 1 all the way to 19, and then chromosome X. And so here what we found is that there's regions of chromosomes 18 and 19 coming together, chromosomes 2 and 4 and 5 coming together, but the red and the blue interaction hubs never interact. They're always separate. So what are they? Upon further inspection, what we found is that this blue one represents the inactive hub, in that they are enriched for genes that are always turned off, or for the most part are turned off. Versus these regions that are in the red correspond to what we're calling the active hub, and these are typically enriched for genes that are turned on. But what are some of the other molecular hubs that we found? So this is just in the context of DNA, but because I told you that DNA is copied into RNA, and because we could extend our Sprite procedure to include RNA molecules, we started to understand how RNA-RNA hubs actually engage with each other. And so that's what I'm going to show you next, in that we revealed not only these 2 DNA interactions, but many other RNA hubs. So this is a heat map that shows orange are molecules that are interacting close with one another, and blue means that they're not interacting very close with one another. And so I'm just going to highlight a couple examples. One of them is the nucleolus. So we actually pull down the entire nucleolus, and when we sequence the RNA, we find that a lot of the RNAs that are within the nucleolus correspond to locations that are very close to one another, as you might expect. And all these other different hubs that I'm not going to get into, but suffice it to say that we found these multi-way hubs of RNA and DNA locations throughout the nucleus. And just to give you a picture of what this looks like, we had a pretty good idea of specific molecules that were in the nucleolus or the nuclear speckle, the Hall bodies, but we didn't have the full picture of every single molecule that exists within these compartments. And because of this unbiased sequencing approach, we were able to discover dozens of unidentified interactions between RNA and DNA within these different membraneless bodies. So this was really exciting because now we have a list of RNA and DNA molecules that exist within these different compartments, and we can start to test hypotheses of what happens when we look at those locations or what happens when we perturb it. And so I'm going to focus on one example, and this is the spliceosomal hub, which I will show you corresponds to the nuclear speckle. And that leads me to the second part of my talk, which is on function. So why do these maps matter? Why is it that the genome has to be organized in this way? And for that, I'm going to give you a brief introduction into how information flows through the cell in terms of genetic information, and that is the concept of the central dogma of molecular biology. So this is the nucleus of a cell with all of our DNA. It gets made into a copy of RNA. This RNA then engages with a ribosome, the thing that makes the protein, and now you have a protein that's made from the message that's contained within messenger RNA. The protein can do all the things that it needs to do for you to function normally. But there's one additional step that is critical in the production of mRNA, and that is a process called mRNA splicing. So before you have a happy, jolly mRNA that's going to produce a protein, it exists as this long ribbon of nucleotides as a pre-messenger RNA. So it has these exons, which are the meaty parts that we want, and these introns that are kind of the junk that we want excised and removed. And so in proper splicing, you have the exons that are stitched together into a mature form, and then this goes out into the cytoplasm and gets made into a functional protein. But what happens when this goes wrong? Well, when this goes wrong, what you can have is improper splicing where the intron didn't get cut out. So now this is a problem for the cell, and it executes different things. So one way that it could react to this improper splicing is that it degrades the mRNA altogether, and you don't get the protein that you need to make. Not good. And so this is the basis of some diseases. And then you can also have a defective protein. If the message actually gets translated and the ribosome's like, I don't understand this, but I'm still going to make a protein, that happens too. And so these have been associated with a whole host of different diseases, including the ones you see here: neurodegeneration, cancer, developmental disorders, immune dysregulation. They are often marked by mutations in spliceosomes, splicing factors, etc. Therefore, for this part, it's very important for us to ask the question, how does the cell ensure rapid and efficient splicing? Because if the cell isn't able to do it, it can't make the proteins it needs to make, and it'll die. So this is not a new concept. This is something that has been thought about for several decades now, since the discovery of splicing, which was 35, 40 years ago. And so I'm going to walk you through what people have gathered about what is the way that mRNA splicing is efficient. And so what people have thought is, okay, so here we have a piece of DNA, and then we have this enzyme called polymerase that finds this location on DNA, and it starts synthesizing this long ribbon of nucleotides, this pre-messenger RNA. Now, where does this enzyme, the spliceosome, come into being? Well, there's this region on the polymerase called the C-terminal domain, name not important. But what is important is that it now acts as a landing pad for the spliceosome, the enzyme that executes the splicing reaction. So now you have the spliceosome and this ribbon of nucleotides directly near each other, such that the spliceosome can now deposit on the pre-messenger RNA, cut it up, take out the intron, and then execute the splicing reaction just fine. So now you have a messenger RNA that's fine and ready to be made into protein. And so in this nice way, you've coupled the production of the RNA with the processing of RNA. So it's like an assembly line. While you're making it, you're processing it, and then the cell can do what it needs to do. And this led to some questions about, well, where are these spliceosomes? Where are these splicing factors in the nucleus? Aha. Well, it turns out that a lot of these spliceosomes were in nuclear speckles. So this is just a cell nucleus here in blue. The red is the nucleolus. This is the Cajal body in white. And then there's these 20 to 25 tiny, teeny little dots known as nuclear speckles. And these actually have been known for several decades to include splicing proteins and the same non-coding RNAs that we found in our sprite maps. And so this was really interesting. And what people thought when speckles were first discovered is that because they contain all these splicing proteins, they represent the site of splicing. So they thought, okay, this membraneless organelle represents the site of splicing because they have spliceosomes, which is a fair assumption. But instead what was found is that spliceosomes exit the speckle and they go to sites of active transcription, the synthesis of RNA, where they execute the splicing reaction. In other words, speckles are warehouses of inactive splicing factors, and splicing doesn't happen at the speckle. So that is a view that's held until today. But, so speckles have long been thought about as RNA and protein, but I just told you that we have this entire new atlas of genomic DNA and how it's organized. And so this is the view that I showed you where we have these active hub DNA regions. And now I'm going to tell you what this active hub is. And what we found is that it's actually the DNA that's organized around nuclear speckles. So let me walk you through what I mean by that. Let's take this long chromosome. And so there's these two genes, gene 1, gene 2, and they're separated by a very long genomic distance, 10,000 kilobases. As a reference point, just an average gene length is like just 10 kilobases. So this is very, very far apart. They don't really see each other if we can humanize them a bit. And so what we found is that if we look at these speckles, this DNA actually folds in such a way that these two regions that were really far in linear distance actually three-dimensionally come around the same nuclear speckle. So which are these active genes that contact nuclear speckles? And for this, I'm going to go through, the next slide is highly technical, so if you don't follow it, that's okay. But it's for any of the biology aficionados for me to showcase how we can use our Sprite technology and how we analyze it so that we can get to these conclusions. And so the way that we visualize our Sprite data is by plotting a histogram of what we call our speckle hub contact frequency. So this is how frequently does each genomic region contact a nuclear speckle. So this is chromosome 10, 41 megabases. That's 41 million bases of DNA. And then these are two regions that are zoomed in. And so a higher value on this histogram means that it's speckle close. A lower value means it's speckle far. So as an example, this is a speckle close region because it has a high speckle contact frequency, and this is a speckle far region because it has a low speckle contact frequency. Well, we can look at these same regions by imaging, and we can image by microscopy because we have methods to do this. We can look at the speckle protein. This is just one of the speckle protein names, and then we can also add a fluorescence probe to the DNA sites that correspond to these two regions. And what we find, again, this is a nucleus of a cell, and then in white are the different proteins, and in blue, which is this TCF3, close to the speckle, this gene far from the speckle. So in essence, it's basically showing us what we're seeing in Sprite. And when we look across the entire genome, we can plot on the y-axis the distance of each and every gene from the nearest speckle. And then on the y-axis, we can plot our Sprite speckle contact frequency. This is all to say that we can actually see that there's a very high anti-correlation where the closer you are to the speckle in Sprite distance, the closer you are also in this microscopy-based data. The bottom line here is that Sprite is accurately measuring our distance to the nuclear speckle. So that's really cool. And so now the question is, well, I told you that these active genes are around the nuclear speckle. The question now is it just simply that a gene, it being turned on, does that mean that it goes to the speckle? Is it all active genes? There's a lot of active genes. Well, the short answer is no. Not every active gene goes to the speckle. So what's the flavor of a gene that goes to a speckle? Well, here I'm showing you, again, low contact frequency, high contact frequency. So this is a zoom in of equivalently length bins for chromosome 7. And then now I'm going to show you these two regions and how much they're transcribed or how much RNA they're producing. And what we find is that, okay, here there is a gene that's on, but it's not close to the speckle. So then what's unique about this site that actually does make it close to the speckle? It turns out if we look at how transcription is occurring there, there is a lot of transcription that's occurring across the entire block. And so there's an increased density of transcription or increased number of genes that are turned on in the similar length scale. And these genes are the ones that tend to be associated with nuclear speckles. And this is just to show you that this is correlated very well genome-wide, where we look at the density of transcription versus the speckle contact frequency. Again, correlated, and these are not just cherry-picked examples. So what this might look like in the context of this of speckles is that if we zoom in at a speckle, So here is our speckle here. So regions that have very little amount of RNA, they still are being transcribed, but they're just not going to the speckle. But regions that have a lot of transcription, a lot of RNA, tend to be more associated with the speckle. So what I've showed you so far is that there are these actively transcribed genes, and they're around the speckle, and there's also active transcription that happens away from the speckle. It's just less. All right, so I started this part by telling you that speckles are these warehouses of inactive splicing factors. But now I've also told you that there's these increased number of genes that are at the nuclear speckle. And so this gave us a really interesting idea of now we have a list of genes that actually contact the speckle. So can we look at the genes that contact the speckle in our sprite data and ask, are the splicing of those genes in any way affected? So does organization increase splicing efficiency of those genes? That's what we wanted to test next. And to introduce that, I want us to take a step back, because I know I've shown a lot of data. And I want to introduce about just chemical reactions and what they depend on. So now we're going to just briefly illustrate, OK, so you have a reactant that goes to a product. And there's an enzyme in between that catalyzes this reaction. And the other thing that reactions depend on are pH and temperature, which are relatively constant in a cell. So that I'm going to remove from the equation. So in the context of splicing, our reactant is our pre-messenger RNA, our product is our mature messenger RNA, and our enzymes are the spliceosomes. And so what I just showed you is that instead of these genes being all throughout the nucleus, there is an enrichment of what we're calling these. So these are the reactants, the pre-mRNAs. And they actually are localized at the speckle. So that means that they have a high concentration of reactants at the speckle. So now the next two questions are, for us to actually complete this model, we need to demonstrate that the spliceosomes are concentrated also at these genes, and that the product is also increased. So that's what we're going to go next. So this result might seem a bit trivial, but it was an important one, which I can't get into the specific details, but what we can do is we can ask, what is the concentration of spliceosomes at these specific regions? And so the way we do this is we look in our sprite data, and I told you that we use this cross-linking agent called formaldehyde. It creates these cross-links between all these different spliceosomes, as well as the pre-mRNA and the DNA. So we have this entire molecular hub that we've captured and frozen. And so now I can measure how many spliceosomes are engaging with this pre-mRNA. So I can look at a speckle close and a speckle far region. And on the x-axis, I can map basically the spliceosome concentration, which I'm calling snRNA density. And what we find, these are 4 different RNAs that are found within the spliceosome. And they are all enriched at the speckle and not enriched when they are at genes away from the speckle. So what this looks like is I began by telling you that there's active genes and these pre-mRNA reactant molecules are around the nuclear speckle. And now I'm showing you that the splicing protein concentration is also highest near the speckle, and it decreases as you get further and further away. What this looks like at the level of an RNA is that if you have 5 RNA molecules far away from the speckle, there's less spliceosomes available. So you're going to have the hypothesis you'll have less splicing, versus up here you have high concentration of spliceosomes, same number of molecules, but they're all engaging with spliceosomes. So that's basically what I showed you here. And so now we have an increased number of enzymes. Great. So now this is a very interesting biochemical problem, which is if we concentrate reactants and enzymes, does this in fact enhance splicing? The way we did this is by isolating RNA molecules as they're associated with the speckle through a method called nascent RNA sequencing. I'm not going to get too much into the nitty-gritty of how we did it, other than to say that we have to do a very stringent biochemical purification method in order to strip away all other RNAs, the other 99% of RNAs in the cell, and we just pick out the RNAs as they're close to the speckle. And so we then can do next generation sequencing and sequence the RNA molecules and ask what is their splicing state, as in how many of these molecules are spliced and how many of these molecules are unspliced, as in they have introns, unspliced. And this will give us a measure of how effectively this process is happening of splicing. And the data will look something like this, where if you have, this is the intron region of a gene and this is the exon. So a region that has low splicing efficiency would mean that there's a high number of introns, versus a gene that has high splicing efficiency would have a low number of introns. So pretty simple. Well, how does the data look? Now, here I'm showing you the Sprite speckle hub contact frequency. This is just to show you that this is a far region, and this is a close region because it has a high value. So how does the splicing look here? Well, when we look at this nascent RNA, this region has a lot of introns, meaning it's low percent spliced. And in other words, it's splicing that's not happening very efficiently. Versus this gene that is located within the speckle has very low number of introns reflected by a higher splicing efficiency and therefore proper splicing. And again, to show you that these are not cherry-picked examples, when we look across speckle close and speckle far regions, and on the x-axis we're plotting the percent splicing, speckle close regions on average have twofold higher levels of splicing than do speckle far regions. And when we look across the continuum of speckle hub contact frequency, there is a nice correlation between speckle hub contact frequency as well as splicing efficiency. And this is all to show you that genes are exhibiting different splicing efficiencies based on speckle proximity. Great. So what I showed you is high concentration of spliceosomes and reactant molecules at the speckle, more engagement at pre-mRNAs, the splicing reaction. The details here do not matter, but basically you're getting spliced mRNA at both locations. But what that means for the cell is that it's low efficiency far away and high efficiency close to the speckle. So indeed, we're finding that there's increased amount of mRNA product when you get to the close to the speckle. So we've kind of completed this entire picture of more reactant, more enzyme, more product. The rich get richer is kind of the motto here, unfortunately. Well, we wanted to take it one step further, and that was we wanted to decouple genomic position and speckle proximity. Can we get cause and effect? And the way that we wanted to address this is, can I produce the RNA anywhere within the cell and drive it to a speckle to drive it to get spliced? That would demonstrate that speckles are actually explicitly coordinating splicing. So how did we do this? Well, the way that we did this is by recruiting different, like one specific RNA to the nuclear speckle. So this is a speckle protein called SRM1, and we fused to it a fluorescent tag M-cherry, which fluoresces red, and so we can visualize it by fluorescence microscopy. And then we also added within the same cells this mRNA reporter, which contains a stem loop structure called an MS2, and this, all you have to know is that when you put them in the same cell, this MCP protein actually binds with high affinity to this MS2. So it's just a way for us to bring the RNA to the speckle. And to show you that this works, when we do imaging for this RNA, we show that the speckle protein co-localizes with the RNA very well. And this is an important check because many times in biology, and anyone who's in science here will know that sometimes things don't work. So you have to make sure that everything is looking spick and span, which is why we have controls. And the control here that we had is a protein that localizes to the nuclear periphery called laminin binding receptor. So it has a very different localization pattern. So we would expect that the RNA should also go there. And indeed, as our control, we do see that the RNA is going where the protein goes. Great, so our system looks like it's working. Next, we want to see what is the splicing efficiency. So when we put all these molecules together in the cell, only if this intron gets cut out, spliced out, will we have this green fluorescent protein be made. So now we can measure green fluorescent protein as a proxy for splicing levels. How does the data look? Well, we didn't only make just the nuclear periphery and the speckle protein, we actually made different classes of proteins that the RNA will get recruited to. And that also includes splicing proteins like these ones, SRSF3 and 9. These localize in a diffuse pattern throughout the nucleus of a cell. And there's another protein, SRSF1, which exists in a dual state, where it's both compartmentalized and it's also diffusely scattered throughout the nucleus. So it'll be important in a second. And so we basically generated 2 versions of this, one where we can actually recruit the RNA to the domain, and one where the protein doesn't have the recruitment domain. And then we can measure the change in GFP, so basically the y-axis is the splicing efficiency when we recruit to all these proteins. So just to orient you, when we look at the nuclear periphery, for all concentration regime of LBR, we see that there's no difference in splicing when we recruit to it. Not surprising, because LBR is not involved in splicing. Well, what happens when we recruit to these other splicing proteins, but they're not at the speckle? Again, there's not really any difference in splicing efficiency. Next, when we recruit to this speckle protein SRSF1, we find that there is a marked increase in the levels of splicing. And then finally, we recruit to SRRM1, which again is localized exclusively within the speckle. And when we recruit to SRM1, we see a over 30-fold increase in the levels of splicing. So that's pretty remarkable in that pre-mRNA localization to a speckle is sufficient to increase splicing efficiency. And this suggests that there is a causal role in organization around the speckle and splicing occurring. And that wraps up that part of that talk. And I just want to impress upon you that I began my talk by introducing all of these different membraneless bodies within the nucleus of a cell. And although we've very comprehensively identified how the nuclear speckle functions and how the structure is important for its function, we can now use this as a paradigm to understand whether the efficiency of the processes that occur within these other membraneless compartments is also the same paradigm that we've discovered for the speckle. And finally, I'll just provide one short slide on what applications I'm thinking about based on these results. And so I showed you this slide earlier on where if you have proper splicing, you make functional protein. If you have improper splicing, you can have various negative outcomes. And you might imagine that there are instances where making more functional protein might be very advantageous to either someone with not enough protein, or even just in like vaccine development where you need to make copious amounts of protein. And decreasing improper splicing would also be a very important thing to do. So one idea would be to take any gene that may be not being as well expressed, and you could recruit it to the nuclear speckle just so that you can increase the amount of protein. And so there's quite a few diseases in which you have what's called haploinsufficiency, where you're not making enough protein. And by recruiting to the speckle, we may be able to start to understand how we may be able to modulate splicing around speckles as like a new therapy. And that completes all 3 of those parts. And so I showed you how we developed Sprite to map DNA and RNA organization, and we made some of the highest resolution maps of 3D organization in the cell. We then talked about how DNA organization around nuclear speckles increases splicing efficiency. And then finally, just very briefly, I just mentioned some implications, how we can think about this for enhanced protein synthesis. Lots of people to thank. I just want to specifically mention a few people who have been instrumental for these projects. Of course, my advisor, Mitch Gutman. He's been an incredible source of support and ideas. He's just a very awesome guy. Open door policy, can always go to him for anything. And it's been a pleasure to work with him. Amy Chow, Ben Emmer, Olivia Etlin have helped a lot with the MS2-MCP assay you saw. And Sophie Quinodoz for the first half of my talk. It was her PhD project that she started with to generate this technology called Sprite. And so a lot of the structural work was done by her. And everyone else that's been on the project, Ina for the beautiful illustrations you saw in her talk, my funding sources, and all of you for your attention. Thank you so much. And do we have time for questions? Yeah. Questions? Yes. Surya.
Again, this was so much work, it was hard to keep up. And again, congratulations on defending. I have a quick question about something you mentioned earlier in your talk, which was we don't know how that if you soup subnuclear components, subnuclear organelles, you get function. And so I wonder if some of your further studies have added more clarity on that question.
For the other bodies? Or?
So you mentioned that if you soup together ribosomes, you don't get protein, but if you soup together subnuclear organelles, that you do get expression of ribosomes. Does Sprite and your research, following research, subsequent research, did that add any more details?
Yeah, so the question was, like in the beginning of my talk, I was talking about how you can kind of execute a lot of the reactions that are within these compartments inside of a test tube, and that here we demonstrated that organizing around these compartments is important for their functional efficiency. And so one of the things that I've thought about in the context of that is when we look at these older papers, so for example, for the nuclear speckle, if you take the nuclear speckle components out and you put it in a test tube, you can still get splicing inside of a test tube. But what people weren't looking at was the efficiency. They were just looking on like an agarose gel, like basically it was a binary, like do I splice or do I not splice? It wasn't what percentage of splicing do I get, and it was in a very synthetic system. So I think a lot of those experiments were important for understanding the biochemistry of splicing, but not really important for understanding like what it means in the context of like in situ in the cell. Ellen.
Really nice job, Prashant. I'm delighted to see this. Can I ask you a scientific question about biology? As you know, and you went over this very lightly for good reason, but you know that there are a number of messenger RNAs that are supposed to be spliced alternatively, and where sometimes the preference for one form versus another is variable. I was just curious if you looked at any of those loci to see if bringing them up close to the
Mm-hmm.
to this speckle was actually overriding that very fine control, or whether it was actually maintaining that kind of cell type and cell activation control.
So yeah, so the question is that right now, so I was showing that we were looking at a very specific type of splicing, and that is intron retention, where whether or not an intron is retained. But there's other types of splicing reactions that can occur, and one of them is alternative splicing. So you can have 3 exons, and 1 and 3 get spliced, 1 and 2 get spliced, and so they can do different combinations. And so the question, I think, is have we looked at whether or not that might be influenced by proximity to the nuclear speckle? It is an amazing question that for our Sprite data, because of our resolution, our read lengths, we can't really answer a lot of the questions about whether alternative splicing happens like close to the speckle versus not. But we can look in some of our nascent RNA sequencing data, and we are looking right now to see if there are differences in alternative splicing. Yeah. Yeah, Lambda.
Congratulations, and you did a great job with this presentation. It's super cool. But so I have a question. So you mentioned like diseases where you like haploinsufficiency, where you don't make enough protein from the gene. But what if you have a disease where you make too much of a protein? Well, maybe not as dramatic as like oncogenes in cancer, but like more subtle. And so can you like unrecruit those genes from the speckles?
Yeah. So. That's an interesting question. So like Landa's question was that I showed that you can recruit to speckles as like a clinical strategy in order to enhance protein production, but can we do the converse of moving it away from the speckle to decrease protein production when you don't want it? Yeah, so I think my immediate gut reaction is that does make sense, but one thing is that not all genes are located at the speckle. So we would first have to identify that the gene that you're interested in that is expressing at very high levels is indeed at the speckle, and if it is, then you could see that as a viable target for disrupting that interaction. So there are some genes, as I mentioned, that are far from the speckle that are still on and they're expressed, and not all genes are at the speckle, so that would just be the one caveat. But yeah, it's a great idea. Yeah.
wondering if there's alternative ways to increase splicing efficiency, like just overexpressing the spliceosome, and if recruitment to speckles offers advantages over these alternative methods.
Yeah. So for the first question, your advisor, Michael Alowitz, also asked me this question, which is a very good question. And are all speckles the same, and do they vary by cell type? And the short answer is we don't really know, and we would love to test that question. And so speckles, of course, are demarcated by many different proteins. And so right now our Sprite technology is able to measure DNA and RNA, but not really great at measuring protein. But there's other methods like Seqfish that Yodai Take has developed, where we can start to look at DNA RNA organization around different speckle proteins in different cellular systems and start to answer that question, but that experiment hasn't been done yet. And the second question you had was regarding other alternative mechanisms like increasing splicing protein production as a mechanism to increase splicing efficiency. So there are diseases in which you have upregulation of different splicing factors, specifically in the context of cancer, and you have rampant splicing. And I don't know if there's a cause and effect here. I don't know if it's a consequence of disease, but my hunch is if you now increase the global concentration of spliceosomes, you've now increased the level of spliceosomes at regions that are far from the speckle, and you're going to aberrantly splice things that shouldn't be spliced. So you need to have a tightly controlled buffer system where you're controlling splicing where it should happen and when it should happen, and not, and because biochemical reactions depend on the enzyme and substrate concentration, by globally increasing, you're going to increase everything, non-specifically. So that would be a little scary. Yeah.
There must be some biochemical reactions that attract, like you mentioned that two genes that are 20 kilobase pairs, 20 megabase pairs apart, but they can come into contact around the speckle. Is there some specificity for gene loci and speckle? And what is the biochemistry of attracting different genes towards a certain speckle, and whether what are the contents of a speckle? So maybe I missed that.
Yeah. So. The question was what are the contents of a speckle? What determines whether two genes end up at the same speckle? So I didn't get a chance to show this data, but what we as well as Yodai and other folks have shown is that most speckle contexts between different cell types do not change. So the same genes are in most contexts contacting in a conserved way. But the things that are really important are that genes that tend to be really highly expressed in different cellular contexts. So for example, in a muscle cell, you make a lot of MyoD1. That is far away from the speckle in an embryonic stem cell, but then it moves closer to a speckle in the context of a muscle cell. So that's data that we have that we did with Barbara Wold's lab. And so basically what we're seeing is that it's transcription of the gene that's toggling a gene either to or from the speckle. But yeah, about 80% of speckle contexts are conserved between cell types. Yes, Nicolas. Thank you.
I'm wondering about, could you go back to your slide where you have your contact mapping where you show the two hubs? So you have interactions there between these two regions.
It'll take me a bit to get there, but.
Yeah. Where they mostly are between certain chromosomes. And then there are other chromosomes for which you didn't find any interaction. So why is that? Or does that depend on a threshold that you use for the analysis? What's happening with these other?
Oh yeah. So so the spider plot that I showed, the circle with the two different hubs, there was the blue and the red, and so there were some chromosomes that didn't have any contact. So that's a very astute observation. So again, it was a little bit of a simplistic. So when we I'm going to answer this technically because I know that you're familiar with it. But basically what we did is we have this interchromosomal contact score where we know that these three different trios of chromosomes interact with one another. And if they interact in a trio, which means a higher order, more than 2, then we call it as an active hub contact. But not every chromosome has that. And so what we then do for the speckle hub contact frequency, which is a separate metric, we divide the entire genome into equivalently spaced bins, and we ask, how does this bin contact one of these regions that I showed you on the spider plot? And then that's how we get a distance to speckle. So the regions that were like not in the spider plot, it's not that they're not near the speckle, it's just that they don't participate in a like multi-way contact, at least that we can detect by Sprite.
All right, let us thank Prashant again for a wonderful talk. Before we conclude the session, I would like to make an announcement that there will be a third talk in the lecture series that's going to be happening over the summer in August. The date is still to be decided, so please watch out for our email. And thank you everyone for coming out today to listen to the wonderful talk.